Dear students, now we are going to see about next category is vector. In the previous class we have seen about enzymes, various types of enzymes. Three important tools to be required for DNA recombinant technology. One is enzyme, second one is vector, and third one is host organism. The second part is called vector. What is vector? Suppose if you want to come to school, you should leave from home to school. You need a vehicle to transport from your home to school. So what is the vehicle to be needed? Either you can come by school bus, or you can come by bike or car or anything else. So the vehicle which are using by you to transport you from home to the school. So this is called a vehicle. Now we are using a same material or same type of word we are using, a cloning vehicle or cloning DNA or vector. So suppose we have joined this plasmid with gene of interest. This have to be transmitted from one place to other place. That means the gene of interest to be transmitted from one place to other place with the help of a vector. That vector is nothing but this plasmid. Suppose this is gene of interest. This gene of interest is pasted, then only this will be transmitted to the host cell. So now the plasmid is acting like vector or a cloning vehicle or cloning DNA. So these are available in some bacteria, some of the yeast cell. These are having this type of, most of the bacteria are having this circular DNA. These are extra chromosomal DNA. These extra chromosomal DNA are called as plasmid. These plasmid are used as vector. These are, some of the vectors are available naturally. Some of the vectors are cultured in laboratory. So the details of this vector we can see. The formation of recombinant technology can be done with the help of this vector. See here, this is the plasmid. This is the gene of interest. This gene of interest to be transmitted to the host cell. This gene of interest to be need for us. This gene of interest is going to give a new product. In order to get this gene of interest, this gene of interest to be carried to the host cell with the help of a vehicle called plasmid. This is called as vector. This is called as cloning vector. This is called as cloning DNA. Now, with the help of the restriction enzyme, we can cut this plasmid as well as the gene of interest here. After cutting it, this will be removed in such a manner. Similarly, we can cut the gene of interest as well in the similar fashion. Then we can join with the help of enzyme called DNA ligase. Then it is pasted. Now it is recombinant DNA is formed. So, in order to transfer this foreign DNA, we need a vehicle. This vehicle is called vector. This vehicle is called plasmid. So this is we have seen now. So now we can understand about what is enzyme, what is vector. What are the properties of vector? Vector should be having certain property because this is a foreign material. This foreign material to be transported into the another cell. That is only recombinant technology or genetic engineering. So the vector should have some basic needs. What are the basic needs? These vectors are able to replicate automatically to produce multiple copies of themselves along with their DNA insert inside the host cell. What do you mean? After this vector is inserted into the host cell, they have to be multiplied themselves because we need more amount of the gene of interest product. In order to get most amount of gene of interest product, they have to be multiplied themselves. That is a very important thing about vector. Another thing, the vector should be very low molecular weight, which should be less than 10 kb, kilo base pair. So in this size, which is easily we can able to transmit. Another thing, the vector must contain origin. What do you mean by origin? Origin of replication. Replication means which can multiply by themselves inside the host cell. So after transmitting this rDNA into the host cell, they have to be multiplied as many number are possible. Millions and millions of cells to be reproduced. This is called, what do you mean? Replication. So replication means multiply in more number. So they have to be multiplied with themselves. So auto replication. So that is, in order to do the auto replication, there is a gene is necessary, which must be present in the vector. That is called origin of replication. And it is also should have a marker gene. What do you mean by marker gene? The marker gene which is having antibiotic resistance to permit its detection inside the host cell. Why do we need this antibiotic resistant marker? Suppose if you transmit this plasmid into the host cell, if the plasmids are having a resistance against this plasmid or rDNA, what will happen? This rDNA will be damaged or destroyed. So in order to protect this rDNA by themselves, there is a marker gene or antibiotic genes to be present inside the vector or plasmid. Another important thing is, vector should have unique target site for integrations of with DNA insert and should have ability to integrate with DNA insert. So every vector to be adapted to fix the gene of interest along with the DNA. This is most important. Otherwise, the total thing is failure. Sometimes only one cloning site is there. That means only one restriction enzyme can able to cut the plasmid. But most of the occasion, many site will be restriction site. Restriction site means what? The restriction enzyme can cut the DNA at as many number of possible places, that is called as sites. So these are called as restriction sites. So if it is having more number of restriction sites, these are called as multiple cloning site. That is called MCS. Or this is also called as polylinker. So presence of this multi-cloning site, that is MCS, facilitate the use of restriction enzymes or choice. So lot of restriction vectors will be available, depends upon which type of MCS we are available. On the basis of it, we can select the vector. So 3 important things to be needed in every vectors. They should have origin, that is origin of replication gene, and selectable marker. Selectable marker means which are having anti-resistant genes against the host cell. Because if the host cell are having antibiotic material, then immediately the rDNA molecule will be destroyed. So in order to protect from them, what is the gene to be need? Marker gene. The marker genes are nothing but anti-resistant gene. So as many number of cloning sites also might be there. So this is the properties of vector. Next we are going to see various types of vector. Natural vectors are there, or synthesize the vector which will be produced in the laboratory are called as synthesis vector, or plasmid are also produced in the laboratory. So in the earlier picture we have seen what is plasmid. Plasmid is nothing but inside the bacteria we are having extra chromosomal DNA. Apart from the regular hereditary chromosome, hereditary DNA might be there. Apart from this hereditary DNA or chromosome, the extra chromosomal DNA which will be circular or double stranded circular form which are present inside the bacteria are called as plasmid. This plasmid will be used for DNA recombinant technology. So here, so this is a bacteria. This bacteria is having bacterial chromosome as well as the extra chromosomal double stranded DNA. This is called as plasmid. This plasmid we can use for DNA recombinant technology. This is natural bacterial DNA which are present in the E. coli bacteria. The second one, PBr322 plasmid. So this is the reconstructed plasmid. That means this is artificial plasmid. It is not natural plasmid. It is a reconstructed plasmid which have been produced by various scientists. Bolivar and Rotix, which have been, these are the scientists who produce this plasmid. This plasmid is most useful in regular DNA recombinant technology. So in this PBr322 plasmid, the P stands for plasmid. The next two letters, capital letter B and R, which will be denotes or which will be named for the scientist, namely Bolivar and Rotrix, who discovered this plasmid or reconstruct this plasmid. Their name is Bolivar and Rotrix. What is 322? The number 322 is a number of plasmid developed from their laboratory. These two scientists have been prepared more than 322 plasmid. Among them, this is 322nd plasmid have been discovered. So these are also contain origin. That means origin of replication. So these are having origin. And it is also having a marker. Two markers are there. What do you mean by marker? Resistance gene. Antibiotic resistance gene. There are two different. Ampicillin resistant gene, tetracycline resistant gene. There are two resistant genes are also there. As well as various sites. What is a site? Restriction sites, which are having various types of enzyme. Hind third, E. coli, BOMH1, SAL1, PVU second, PST1, CLA first. These are all various types of restriction enzyme which are having various restriction site. That means MCS. MCS means multiple sites are available. So these are very much useful in modern day technology of recombinant technology, that is genetic engineering. So PBR322 plasmid is a reconstructed or man-made plasmid. Next we are going to see about TA plasmid. This is a natural plasmid. These are available in normal soil. So there is a bacteria, Agrobacterium tumefaciensis. This is a natural bacteria which are available in the soil. As well as these bacteria, it is available everywhere else. So this is having a specialized plasmid which are called as TI plasmid. TI means tumor inducing plasmid. Tumor means cancer. In plants which can cause cancer with the help of this gene called TI gene. That is why this plasmid is also called as TI plasmid, tumor inducing plasmid. So this is available in a bacteria called Agrobacterium tumefaciensis. So this bacteria, which will be responsible for lot of genetic engineering. I mean, natural genetic engineering may be occurred due to with the help of Agrobacterium tumefaciensis. So this is also can cause tumors or cancer in several dicot plants. This plasmid carries transfer gene, which can helping to transfer the DNA. So these are also called as tDNA. So transfer gene is available in this plasmid, which will be helping to transfer the DNA from one bacteria to the another bacteria, or one cell to the plant, one plant cell to the another plant cell can be done with the help of the gene called transfer gene. It is also having ONC gene. ONC means oncogenicity. Onco means cancer. Oncogenesis means cancerous property. So the cancerous property which will be done by this plasmid, this gene is called TA or oncogene. So the oncogene is responsible for oncogenicity, which are also having origin. Origin means origin of replication. And it is also having various genes like INC gene. INC gene means incompatibility, which are not suitable, they won't be allowed. So only certain DNA or certain particle can be allowed. Such a type of gene is called INC gene, that is incompatibility gene, which are also present in the TA plasmid. So these are all present inside the same plasmid itself. This is natural plasmid. This is also being used in various types of plants for transferring DNA. Next one is called transposon as a vector. So this transposon vector is also called as walking gene or jumping gene. So transposons is a transposable element or mobile element or DNA sequence able to insert itself at a new location in the genome without having any sequence relationship with target locus, and hence these are called as transposon, or which are also called as walking gene or jumping gene. They are used as genetic tools for analyzing gene and protein function that produce new phenotype on host cell. So that is why these are very commonly used, especially in organisms like Arabidopsis thaliana and bacteria such as E. coli. So this is transposon gene. So this is a gene. This transposon gene is transport into the another sequence without any help of any outer body or any helping. Without any helping, this transposon gene will be transferred. That is why these are called transposon or walking gene. Next one is called expression vectors. The vectors which will be showing the expression of the gene of interest. It is called expression vector. That means what is the characteristic feature which is having by the vector? That character will be available in DNA recombination. So this is called as expression vectors. Then various types of another vectors are also we can go through once. Next one is cosmic vector. What is meant by cosmic vector? This cosmic vector is nothing but they are having cohesive terminus. What is meant by cohesive terminus? These are not having blunt end. These are having sticky end. If it is cut, which is having cohesive end. That is why these are called as cosmic or plasmid containing cos or cohesive terminus. Such a type of plasmids are called as cosmic. Next one is called bacteriophage. This plasmid is called as bacteriophage vector. The bacteriophage means this is a virus which can infect bacteria are called as bacteriophage. So the vector, this gene or the plasmid, if it is obtained from bacteriophage, then this is called as bacteriophage vectors. The most commonly used E. coli phages are 1 phage or lambda phages or M13 phages will be used. These are having 25 kb can be inserted into the phage vector. So that is why these are commonly used. Another one, it is artificial virus vector, that is also called as phage-mate vectors. Phage-mate vectors are constructed plasmid vector which contain their own origin. Origin means what? Origin of replication gene, and also contain origin of replication of phage as well. So this is artificial virus vector. Next one is bacterial artificial chromosome vector. This is also called as BAC vector. So this is also, the BAC is a shuttle plasmid. What do you mean by shuttle plasmid? It is created for cloning large size of foreign DNA. This BAC vector is one of the most useful cloning vector in our DNA technology. They can clone DNA insert of up to 300 kb. So usually 10 kb we used to call, but 300 kb of nitrogenous base can be transferred. So they are very stable, more user friendly. Similarly, yeast artificial chromosome. How? A artificial virus vector, artificial bacterial vector. Similarly, fungus vector, artificial yeast artificial chromosome vector. This is called as YAC vector. So this is also synthesized in the laboratory. YA plasmid vector behaves like a yeast chromosome. That is why it is called as yeast artificial chromosome vector, which occurs in two forms.